Hello and welcome to this module on Lysis buffers. This will be a brief overview of Lysis buffers and the different components that are included within them. So let me begin by discussing the categories of Lysis buffers. We can categorize Lysis buffers into two main categories depending on the presence or the absence of a detergent within the Lysis buffer. So we have detergent Lysis buffers. These have detergents in them. I'll discuss what detergents are exactly in the next slides. But basically, a detergent is an amphipathic molecule that can interact with polar and nonpolar substances, allowing it to facilitate itself in between cell membranes, thus breaking up cell membranes and causing cell lysis. So the other type of lysis buffers is non-detergent lysis buffers. So an example of this would be lysis by osmosis. So if you place your cells in hypotonic solution, that will cause so that water rushes inside the cell and eventually lyses the cell open. When looking at detergent lysis buffers, we can classify those lysis buffers into two main subcategories, being the non-denaturing detergents as well as the denaturing detergents. So non-denaturing detergents are basically detergents that do not interfere with your proteins within the cell lysis or within the cell lysate. On the other hand, denaturing detergents will denature your proteins within the cell lysate. So an example of a denaturing detergent would be SDS. And sometimes we do want to indeed denature our proteins and run them, for example, in a gel electrophoresis subsequently. SDS will not only denature your proteins, but it will also cause them so that they're coated with negative charges such that they can move across the gel towards the anode or positive electrode. So since I started this module by classifying lysis buffers depending on the presence or the absence of detergents, let me begin by discussing what detergents are. So as I said earlier, detergents in general are amphipathic molecules. An amphipathic molecule will consist of a nonpolar group or a hydrophobic region as well as a polar head. So you can look at the circle here as some sort of a polar group. This could be something like a hydroxyl with other polar elements, and the nonpolar part or the hydrophobic region would be things like carbon and hydrogen atoms. So here on the slide, you can see four different types of detergents that are commonly used in cell lysis buffers. For example, you can have the CHAPS detergent. This is a switterionic detergent. Because it is switterionic, it's not as mild as non-ionic detergents like Tween and Triton, but it is non-denaturing. On the bottom, you can see the Tween detergent. Now, the Tween detergent is a non-ionic detergent, and it is also commonly used in cell lysis buffers. And also you have the Triton X detergent that you can see at the bottom right. The Triton X detergent is also a non-ionic detergent that can be used in cell lysis buffers. Now, in comparison to the CHAPS, Tween, and the Triton X detergent, the SDS detergent here is an ionic detergent. Specifically, it's an anionic detergent, meaning that it is a detergent with negative charges, and it will also coat the protein of interest with negative charges. Importantly, the thing to remember here is that SDS is a denaturing detergent. It will denature your proteins within the cell lysate, and sometimes that is exactly what we want, while other times we want to keep the integrity and maintain the integrity of our proteins of interest. Now, it's important to understand that in the case of lysing a cell, it's important to have a lysis buffer that has the necessary components to not only lyse the cells, but also ensure that the lysate is within suitable environment that allows for further and subsequent experimentation. So we said that lysis buffers can be categorized into either detergent lysis buffers or non-detergent lysis buffers. So in the case of detergent lysis buffers, we have detergents that assist in lysing the cell. And in the case of non-detergent lysis buffers, we utilize other means to lyse the cell membranes, for example, by having a hypotonic solution. Now, in addition to having detergents or having hypotonic solutions, it's also important to have other components that will make the lysate environment suitable for the necessary subsequent experimentation. So, for example, one of the fundamental things that must be added to a lysis buffer are salts. And common salts that can be used include sodium chloride, potassium chloride, and magnesium chloride. You'll often see sodium chloride or potassium chloride used within lysis buffer solutions. And there are different reasons for why that is. Now, fundamentally, the overarching reason is to create some ionic strength. And I'll talk about ionic strength in a bit. Now, basically, ionic strength is a measure of the concentration of ions inside the cell. So we know that if we take a look at a cell in physiological conditions, say at a cell within the human body, we know that the cell regulates the concentration of different ions inside the cell as well as outside the cell. So if we take a look at a cell in physiological conditions, say a cell within the human body, we know that the ions within the cell and outside the cell are regulated to have particular concentrations. So, for example, if we were to imagine that we have a cell membrane here and the left side being the inside of the cell, whereas the right side being the outside of the cell, we already know that there are important proteins that regulate ionic concentrations within the cell. And in turn, they also regulate the presence of these ionic concentrations outside the cell relative to the inside. So, for example, if we were to extend this cellular membrane, so if we were to extend it like this, we can draw some of the important proteins that help regulate ion concentrations across the cell membrane. One important protein that you are very familiar with is the sodium-potassium ATPase. The sodium-potassium ATPase will pump 3 sodium ions outside the cell for every 2 potassium ions that it pumps inside of the cell. And so essentially the sodium potassium ATPase is one of the fundamental proteins that help regulate ionic concentrations across the cell membrane such that you have a high ionic concentration of potassium ions inside the cell relative to the outside of the cell, and you have low sodium concentrations inside the cell relative to the outside of the cell. So the ionic concentrations, or the concentrations of ions inside the cell, determine the ionic strength that we have inside the cell. And so one of the functions of using salts in lysis buffers is to maintain physiological conditions, give or take. So the ionic strength inside the cells is roughly 200 millimolars. And what we're doing in composing lysis buffers with salts in them is we are trying to mimic the ionic strength that is inside the cells. Now, importantly, the ionic strength of lysis buffers will vary. So some lysis buffers will have ionic strengths of 50 millimolar, others will have up to 150 millimolars, others will have more, so up to, like, say, 300 millimolars. So the ionic strength will vary based on the cell type that you are dealing with, and also based on what you're attempting to do or what you're attempting to extract. But the idea here is to keep in mind that we are trying to mimic physiological conditions. Now, importantly, increased ionic strength to a certain degree will help solubilize the protein. So when we take a look at the cell lysate here, for example, and let's assume that these are proteins, these dashes are proteins, when you have ionic strength, when you have different ions that are inside the cell lysate, these will keep the different proteins away from one another, meaning they do not aggregate onto one another. Now, ironically speaking, when you have the ionic strength at very high levels, the proteins will actually aggregate in the cell lysate. So keep this in mind. Another component that you'll often see within cell lysis buffers is glycerol. Glycerol is an osmolyte that is used to stabilize proteins within the cell lysate. Now, importantly, the mechanism of glycerol is considered elusive to some degree, but there is some excellent research that discusses potential mechanisms by which glycerol stabilizes proteins inside the cell lysate. This is a particularly good resource that discusses the stabilization of proteins by glycerol. So if we were to look at the figure at the bottom left, this figure is derived from the paper that I just highlighted at the top. And this figure showcases some of the mechanisms by which glycerol can help stabilize proteins. Importantly, on the figure, you can see a free energy diagram that showcases the unfolding of native proteins. Here in the dashed line, you can see a protein with glycerol, and in the solid line, you can see a protein without glycerol, and you can see the free energy diagram of both. Importantly, what you have to the very left of the reaction coordinate is the native state of the protein, and what you have to the right of it here is basically a partially unfolded protein. Basically, you can think of going from the left to the right as unfolding here. And then you can have association where different proteins aggregate with one another. So you can see the protein aggregation happening in here. Now, the idea by which glycerol stabilizes proteins is twofold. So the first idea, or the first mechanism by which glycerol stabilizes proteins as represented in this paper, is that glycerol prevents partial unfolding. So you can see the native state here, and you can see that you have unfolding here. And what glycerol does is that it preferentially hydrates the expanded transition state that you see here. Now, what do I mean by preferentially hydrates the expanded transition state? Well, I mean that it preferentially hydrates this transition state in comparison with the native state that you see here. Because it preferentially hydrates the transition state that you see here is expanded more with glycerol. When the protein is with glycerol, it is expanded more than the native state is expanded here. And because of this, it makes it less likely that the protein will go from the native protein towards unfolding, towards partial unfolding. This is one way by which glycerol stabilizes proteins. So let me explain the other way by which glycerol is said to stabilize proteins based on the reaction coordinate diagram that you see here. So let me change the color of my pen really quickly here and discuss the other way that glycerol stabilizes proteins. So the idea here is that glycerol is also said to prevent the association of aggregation-prone intermediates. So recall that we said that here we have the partially unfolded intermediates. We have the partially unfolded intermediate of the reaction coordinate or the free energy diagram of the proteins without glycerol in the solid line here, and the proteins with the glycerol in the dashed line. What I want you to pay attention to is the following fact. Notice how glycerol interaction with proteins makes it so that it causes a higher transition state that you see here, as well as a higher association, second order association that you see here. Relatively to that, the increase in the free energy diagram for the partially unfolded intermediate is not that much. So this increase here for the partially unfolded intermediate is not much. You only see a little arrow that here as compared to this big arrow that you see here and the big arrow that you see here. Now, because of this, what happens is that these partially unfolded intermediates that you'll have in your cell lysate are less likely to either associate and cause aggregation, but in addition, it is less likely that the partially unfolded intermediates refold into the native state. However, the rate by which the refolding into the native state is decreased is less than the rate by which the partially unfolded intermediate can associate with other partially unfolded intermediates and aggregate. In other words, what I'm saying here is that the rate of first order refolding, recall that when you're looking at a free energy diagram, the reaction can go both ways. So it's natural to think of this going to the right, but it also can go to the left. And so here I'm talking about going to the left when I talk about refolding of the partially unfolded intermediates. So the rate of the first order refolding reaction, meaning the rate of going this way from the partially unfolded intermediates into the potentially the native state. Now this rate decreases. It decreases less than the rate of the second order association reaction. So this right here decreases, the rate of this decreases less than the rate of going towards the association reaction. And this makes it so that it's more likely with glycerol that the partially unfolded intermediates actually move towards the native state than they are to associate and aggregate. So importantly, what you should understand is that the rate of the reaction in the refolding of the partially unfolded intermediates let's say that this is the refolding, which goes this way. This rate of the reaction of refolding decreases. Obviously, it decreases. We can just say rate decreases. It decreases because we have this upward hill that's now been increased relative to the crevice that we see here. And so the rate of refolding decreases. In addition, the rate of the association that you see here, so association, we can say association here, the rate of the association is also decreased, but the decrease in the rate of association is greater. Basically, the rate of the refolding is decreased, but not to a great extent as compared to the rate of the association. And because of this, it's more likely that partially unfolded intermediates will refold as opposed to associate with one another. Now, to the figure on the right, you can see another diagram of the effect of glycerol on proteins and their stabilization. This does not come from a paper that specifically tries to understand the effect of protein stabilization by glycerol, but rather it's also a nice, simple figure to understand. Basically, the idea they try to point here is that you have an unfolded protein without glycerol, and then you have the addition of glycerol. What's happening is that the addition of glycerol is inducing the folding of the protein. The idea here is that the glycerol induces hydrophobic chains found on the surface of misfolded proteins to fold towards the inside and escape from water. And this folding favors the native state. So basically, the idea here is that if you have glycerol with an unfolded protein, this will induce it to fold such that the hydrophobic regions are hidden from the glycerol and from the water, which glycerol attracts to a greater extent. So remember that glycerol is rather hydrophilic, and it's going to increase the hydration of the protein surface. And this naturally induces more folding, and then we would hope that that more folding causes folding into the native state. If I was to choose between understanding the figure on the right and understanding the figure on the left, I'd understand the figure on the left, because inherent to the figure on the left is also the idea that partially unfolded intermediates are more likely to move towards the native state because of the effect that glycerol has on the overall free energy diagram of genes. Now, another component that you'll often see in cell lysis buffers are chelating agents. These are used for chelating divalent ions, and common examples of chelating agents include ethylene diamine tetraacetic acid, or EDTA for short, as well as ectasic acid, or EGTA for short. Now, in EDTA, as compared to EGTA, EDTA will have a higher affinity for magnesium divalent ions, as well as a lower affinity for calcium divalent ions. On the other hand, EGTA, when compared with EDTA, will have a higher affinity for calcium ions and a lower affinity for magnesium ions. So the main function of chelating agents is to decrease the availability of divalent cations that are used as cofactors for enzymes such as DNases and proteases. So you could think of EDTA and EGTA as inhibitors of DNases and proteases within your cell lysate. Now, sometimes within the protease cocktails that you'll utilize within the cell lysis buffer, I'll talk about protease inhibitors in a bit, you'll see that there is EDTA that's been added to the protease cocktail inhibitor. Now, importantly, both EDTA and EGTA can often be used interchangeably, but it is often EGTA that is preferred for enzyme assays, DNA extraction, and protein purification. Now, to the top right, you can see a figure with a chemical structure of EDTA, as well as a structure that shows you how EDTA can chelate divalent ions. Here in white is a divalent ion. Basically, EDTA would interact with that ion and make it so that it is not available for enzymes such as DNases and proteases to use as cofactors. Importantly, when you are using EDTA, you have to be aware of its effect on DNA polymerases. So, for example, if you are lysing a cell using a cell lysis buffer that contains EDTA, and then you are performing PCR afterwards, you have to be aware of the potential effect of EDTA on your PCR, namely your DNA polymerase, your Taq polymerase within your PCR. Now, so this makes it so that you have to use EDTA within a certain amount of concentration or within an acceptable or an appropriate concentration, such that you don't have too much EDTA that can linger and affect your PCR. So recall that EDTA chelates magnesium ions, and DNA polymerases require magnesium ions as a pivotal cofactor. So just to review the DNA polymerization reaction, let's take a look at the figure at the bottom left. So you can see here that you have a sequence, say this is the sequence that's being elongated by DNA polymerase, and you can see that you have the 3' hydroxyl in here. Now the 3' hydroxyl will be deprotonated by a base, and once this deprotonation happens, you'll have an oxygen that's deprotonated that is now more nucleophilic that can attack this phosphate group within the triphosphate of an incoming deoxynucleotide triphosphate. So this makes it so that this deprotonation is pivotal to the process of DNA polymerization. In addition, it also makes it so that the pyrophosphate that will be a leaving group is an important leaving group that also must be stabilized. Recall that when you have a reaction, you want the reaction to proceed such that it forms a stable structure, and the leaving group must be stabilized in the process. So this is where the role of magnesium ions that are in association with the DNA polymerases come in hand. And this is why magnesium ions are used as cofactors within DNA polymerases. So importantly, magnesium ions will play a role both in ensuring that there is relative stability of the deprotonated hydroxyl. So you can see here the deprotonated hydroxyl, and the magnesium ion will be close to this deprotonated hydroxyl and help stabilize this charge. In addition, another magnesium ion will be in association with the pyrophosphate leaving group that will help stabilize it, so that this makes it so that the DNA polymerization reaction is more favorable. So to the figure on the right, you can see the magnesium ions that are associated with the DNA polymerase. So here you can see the DNA polymerase, and the DNA polymerase is within association of this magnesium ion and this magnesium ion that are labeled magnesium ion A and B. You can see that the magnesium ion A is in association with this aspartate amino acid within the DNA polymerase, and the magnesium ion B is in association with another aspartate amino acid in the DNA polymerase. Magnesium ion A is closely associated with the primer, so it is closely associated with the primer that you saw here, namely the 3' hydroxyl on the deoxyribose. I'm circling the deoxyribose right now, is going to be close to the magnesium ion A. And again, remember the base. So when you have a base that deprotonates the 3' hydroxyl on the ribose, then the magnesium ion will in turn stabilize the negative charge on the hydroxyl's oxygen that's been deprotonated. On the other hand, magnesium B ion is closely associated with the pyrophosphate group or the triphosphate group that is incoming on the deoxycytosine triphosphate in this case, that's incoming that will react with the primer. So for example, you can see here in orange and red the triphosphate of the incoming nucleotide, and the magnesium B is closely associated with it so that it can stabilize the leaving group. That is, it can stabilize negative charges on the pyrophosphate that will leave once the primer reacts with the incoming deoxynucleotide triphosphate. So another component that is often used within cell lysis buffers is beta-mercaptoethanol. It is used especially when the goal is to extract RNA, but in reality, it can also be used in other applications that do not involve RNA extraction. And the reason we use beta-mercaptoethanol is so that it can denature RNases. The mechanism of this is the following. It basically reduces cysteine-cysteine disulfide bonds that are prevalent within RNases. And as a result, this denatures the RNases within the cell lysate, and this makes it so that your integrity of the RNA within the cell lysate is relatively more preserved. A fundamental component of cell lysis buffers are protease inhibitors. So when cells are lysed, proteases are no longer regulated metabolically or contained within certain compartments of the cell. And this makes it so that your proteins within the cell lysate are vulnerable to the rampant activity of proteases that are found within the cell. And this is why we use protease inhibitors within cell lysates. Protease inhibitors are needed to prevent degradation of your protein of interest within your cell lysate. Now recall that chelating agents that we've discussed earlier, such as EDTA and EGTA, can also be considered protease inhibitors since they are preventing the activity of proteases within your cell lysate. But often what you'll see is the addition of additional protease inhibitors to your cell lysis buffer that are on top of the addition of EDTA and EGTA. So here to the right, you can see a figure of various protease inhibitor cocktails that can be used in cell lysis buffers. And you can see that many of them, so for example, if you take a look at this cell lysis protease inhibitor cocktail, you can see that it contains various types of protease inhibitors, including EDTA. And you can take a look at the differences between those different protease inhibitor cocktails. And lastly, a fundamental component within cell lysis buffers is in the name. It's the buffer within cell lysis. So buffers include things like tris HCl buffers and HEPase buffers, and they function to obviously maintain the pH. This is essential in protein extraction, but really it's essential in any cell lysis buffer. A good way to think of the importance of buffers in maintaining pH is by comparing it with the importance of maintaining ionic strength by including certain ions within your cell lysis buffer. Both ionic strength and pH are fundamental so that you have an environment within your cell lysate that is comparable to some degree to the physiological environment of the cell. So here, when we're dealing with the cell lysate in vitro, we want to mimic what happens in vivo. And lastly, I want to end with a general note on lysis buffers. So here you can see 3 different lysis buffer recipes that I've included in the slide. And in each of those recipes, you could see virtually all of the components that I've discussed in this module. For example, if we take a look at this cell lysis buffer recipe, you can see that you have a buffer that will regulate the pH. You can see that you have ions that will help maintain ionic strength in the cell lysis buffer so that the cell lysates have a comparable environment to in vivo or physiological conditions. You can see that you have chelating agents that are utilized as components for assisting in inhibiting proteases and DNases. You can see here that you also have different protease inhibitors. So here you have a protease inhibitor, another protease inhibitor, another protease inhibitor. And of course, here you have a detergent lysis buffer. So you have Triton X. This is a non-ionic detergent. You also have glycerol for the stabilization of proteins. And you also have a little bit of a denaturing detergent, mainly SDS, within this cell lysis buffer. So one of the things that I want to bring across in this module is that lysis buffers come in quite a variety. So for example, here you have 2 detergents that are being utilized. In fact, you have 3 detergents that are being utilized. You have the Triton X, you have the SDS, and you have the deoxycholate detergent that I have not discussed within this module. But rather, what I want you to keep in mind is just the general variety that you see in cell lysis buffer. But overall, you have these general components that I've discussed within this module that are present within every single cell lysis buffer, give or take. Here you can see another cell lysis buffer. You can see a buffer here within the cell lysis buffer components to maintain the pH. You have the tris HCl buffer. You also have some ions to maintain ionic strength. You have the glycerol to stabilize proteins. You have the chelating agent, which also acts as a protease inhibitor. And you also have PMSF here, which also acts as a protease inhibitor. Here to the right, you can see another recipe for a cell lysis buffer. You have a buffer that maintains a pH. You have some ions for ionic strength. You have a detergent, namely Triton X, as well as another detergent, which is the NP40. You have another detergent here that is also being utilized, as well as another detergent here, which is SDS, that's also being utilized. So you have multiple detergents that are being used within the same cell lysis buffer recipe. You have chelating agents, and you have sodium fluoride, which is acting as a serine threonine phosphatase inhibitor. And of course, you'll have water in all of these cell lysis buffers. So I think you understand the point that I'm trying to get across. Lastly, I want to end my note on lysis buffers with a remark on the recommendations that are made by companies that sell cell lysis buffers. So you'll often see different cell lysis buffers that are sold by these companies, and they provide recommendation based on whether you're extracting a cytoplasmic protein or a nuclear protein, and whether you are seeking a final protein that will be denatured or non-denatured. So here you can see that you have different cell lysis buffers, and here you can see the different recommendations for different uses. For example, they recommend this cell lysis buffer for extracting cytoplasmic proteins, whereas this for extracting cytoplasmic and nuclear proteins, and so forth and so on. Now, importantly, when you dig deep and look at the various cell lysis buffers that are utilized in different research or different labs, you'll notice that these recommendations are not adhered to stringently. So sometimes a cell lysis buffer that is recommended to extract cytoplasmic proteins may be used to extract a nuclear protein. And this often is a trial and error course. So a lab will try a certain cell lysis buffer. They'll realize that it works for the particular protein or whatever extraction they're trying to do, and they'll continue to use it. So keep this in mind, but also keep the recommendations of reputable companies also in mind. And this concludes this module on cell lysis buffers.